afternoon, good morning, whatever it is, for wherever you are. I suppose most of you are in Europe. And it's a pleasure to be able to speak to you today through the European Astrobiology Institute Academy, which means a way of teaching people about astrobiology, keeping people abreast of the many things that happen in such a wide field as astrobiology is. Today, I will be talking about why and what can you do about answering the why, how to make non-biochemistry-based life in a test tube. Just the title is something that many could probably think that is a contradiction, because people usually associate life with biochemistry. But if you think a little bit out of the box, you don't have to get very far away from the box. If you think a little bit out of the box, you realize that life can be characterized by a series of properties. And those properties, one can ask, can those properties be actually implemented using a chemistry which is not biochemistry? And if so, how and why? So first, the beginnings of the why. In the latter part of the 20th century, in the late 1980s, early 1990s, people knew of the presence of many chemical molecules, organic chemical molecules in the interstellar medium. In the late 1990s and early 2000s, we began to see through space missions, we began to see that the complexity of the chemical landscape in places like, for example, the Orion Nebula is extraordinary. And today we do know that there are at least between 300 and 400 different complex chemical species, and also not so complex chemical species. But in addition to that, and since this is in the interstellar medium, and since 1995, essentially when we meaning humanity, discovered the first exoplanets, we have discovered and seen about 5,000 exoplanets. I forgot the count. The count a couple of days ago was about 5,000, a little bit above 5,000. And we have seen that there are in the cataloged exoplanets that we know, we see that there are water worlds, there are rocky worlds, water worlds, transitional planets. We have gas giants, and so on and so forth. And the variety of exoplanets is so large that it is imperative to start thinking if there is life in those places and if it is going to be based on biochemistry. Why? Because biochemistry probably is the result of the co-evolution of chemistry on planet Earth and the physical evolution of the planet itself. If you're interested in learning more about this classification of planets, you can look at this paper. This is with Li Zheng, a bunch of us as co-authors with him. It appeared in 2019 in PNAS, Proceedings of the National Academy of Sciences. So having said that, so there is all these exoplanets, there is matter in the interstellar medium. What happened on Earth? So we speculate about what happened on Earth between 4.567 million years ago, when our planet formed, and until the last universal common ancestor, 3.6 billion years ago. We speculate a lot about what could be, but there are some signs of regularities that may have taken place. Some are known to have taken place, and others we don't really know the details, but we do know much more than what is expressed in this transparency from Jerry Joyce, which was put together in 2002. Many things have happened. We do know that the planet formed about 4.5 billion years ago, that it was a stable hydrosphere probably earlier than 4.2 billion years ago, that some chemistry could have taken place here. We don't know exactly which kind of chemistry was there, but I would speculate that between 4.3 and 4.0, we definitely had the possibilities of having a lot of chemistry, not not necessarily biochemistry, but a lot of chemistry in our planet, in the surface of our planet, especially after the stable hydrosphere formed. And then it is speculated that there was a pre-RNA world. We don't really know what that is like. And then there is an RNA world, which we know what it is like, but we don't know the details of how it happened, or even how it could actually bootstrap itself. And then we do know that 3.6 billion years ago, there was protein life. And from 3.6 to the present, from the advent of the last universal common ancestor, Luca, to date, everything that we call life is based on biochemistry. There is a region of time between about 4.2 and 4.0 billion years ago, billions of years ago, which we call the protocell era. I like to call it the protocell or generalized biology era. The term generalized biology is a concept introduced by Bernal, an Irish-English extraordinary biochemist and chemist and crystallographer, where he spoke about life, which has all the properties of life as we know it, but it's not based on biochemistry. Similar thoughts were also expressed concomitantly by Oparin. And today there is lots of speculation as to what might have happened there, and if life perhaps started in a very simple way and then evolved into what we know by co-evolution of the chemical systems that we call, that there were then, their co-evolution with the planet, with our planet. The picture that we handle today is that this is a transparency courtesy of Andy Knoll, a colleague of mine here. The picture that we have today is life is today here at the crown of this drawing. We have archaea, 3 domains: the domain of the bacteria, the domain of the archaea, and the domain of the eukaryota. By doing phylogenetic analysis, we can actually go to Luca, as we mentioned earlier, the last universal common ancestor. But we are beginning to think in terms of forebear lineages from before the most recent common ancestor, from before Luca. And that leads one to the question, what could there be here? Was there life here? What was it like? And in addition, asking, was life necessarily as we know it? Like Steve Bernal says, if you don't like life the way it is, make your own. So that's what I decided I would do at some point, because I was seeing very interesting signs happening in my work. And the important point to realize now is that all the above, all that we have seen shown in this, that we see, all the living systems that we see today are based on and use standard physical chemical processes. And those processes, at least since Luca, were implemented with biochemistry. Now, there is a huge leap in complexity from chemistry, or even organic chemistry, or carbon chemistry, to biochemistry, and an even larger, or a comparably larger, I would say, a jump in complexity when you go on to life as we know it. So the first question that one can ask is, does the widely held and validated tenet in fundamental physics that the complexity that we see at the surface of things is a reflection of a very deep simplicity, that which sounds somewhat theoretical in a not very nice way. I'm using the term theoretical in a not very nice way, but that sounds theoretical is actually an extraordinary way to ask questions about complex systems, starting at the outside and then look deep into them. It's called outside in, and this is what we will use for our thinking. So how do we start? So let's look at life in general. Life in general is characterized, as I said today, is characterized by biochemistry, and systems, living systems show evolutionary adaptation, they show reproduction, they show energy processing, regulation, order, and a series of properties. These properties are not the essence of the properties which characterize life. You can actually study these properties and see, as inspired by Schrödinger, many people have done, see that all living systems that we know today, with all these properties and many more that we didn't show, are characterized by the following set of 4 properties. The 4 properties being that every living system uses information for its work, for controlling the chemistry that they perform, for they generate information that they pass on to others that follow them, they transmit information, they store information, they communicate information, and so on. All living systems have another set of properties which are called metabolism, which is the fact that living systems by themselves, totally autonomously, they make their parts and they handle energy, they make their own parts, and then they make parts to actually self-reproduce, which means that the living systems make copies of themselves. And then eventually they, because of the cycle law, because of the growth in entropy, the system will decay eventually, maybe 1 generation or 2 generations or 3 generations, but it will not live forever. And so that is done by the chemistry within the system. Under the control of that chemistry, the system self-reproduces. And living systems are also characterized by the fact that they don't stay the same. They react to changes in the environment. They react to changes in the environment by changing their chemistry, by changing many of the parts that the chemistry makes in accordance to inputs that the system receives from the environment. All of this can be classified in terms of what it's written down. There's evolution, which can be adaptive and non-adaptive. This reduction to 4 properties, 4 basic properties, has been put forth by several people, especially in the last 20 years or 30 years, actually, with Manfred Eigen and Bernd Olaf Küppers. I myself did a little bit of this and studied, compared the previous properties, such as these. I compared them to how they were used to this. And then there is some very interesting papers by Mariscal and Doolittle in the last couple of years. And Paul Merz has written something which actually encapsulates this without entering into any details. And the question that we're asking is biochemistry necessary, or is biochemistry a sufficient proposition? And in order to ask that, we have to ask, can we integrate or unify, whichever word you like better, these 4 functional properties into using water or some solvent, but we will stick to water because that's how life goes on in our planet. So can we create an artificial chemical system that carries out all those 4 properties without using any biochemistry? Living systems, all living systems, execute a very interesting process, which is called the cell division cycle, which consists on an integration of the mass in the system, the information in the system, and energy processing by life. Let me show you an idealized picture of this system, the cell division system. In the cell division cycle, there is, in the left-hand side, you have cell mass, and in the right-hand side, you have a different scale for the DNA per nucleus. So the DNA, the system starts here. The mass begins to grow from size 1, quantity 1, and it goes all the way to quantity 2, at which point it will divide. When it starts growing in mass, it keeps the amount of information constant for a while, and then the amount of information grows, and it goes from value 1 to value 2. And then when it reaches a certain value 2, that value 2, it does some rearrangement inside, and then and this happens before the cell mass is multiplied by 2, and then the system at some point divides and replicates. And this continues and goes on over diverse cycles. This is an integration of all these properties. In science, in physics, for example, we have seen the fact that you can integrate, you can unify many of these many systems. An example which is extraordinary for many reasons is the example of Maxwell's equations. Maxwell set out to combine, to put together electrostatic forces like described by the Coulomb law, and the magnetic forces exemplified here in the Biot-Savart law. So Maxwell put together a set of equations which actually could unify these properties, electricity and magnetism, into a set of 4 differential equations. These differential equations are called, of course, Maxwell's equations. They date from about the same time as Darwin's theory of evolution. This is from 1862. And then he was able to write these equations. And these equations, when you manipulated them mathematically, they actually led to the prediction that there would be waves which were transversal waves. Those waves were discovered about 25 years later by Heinrich Hertz. And Hertz discovered the electromagnetic waves predicted by Maxwell's equations. And then, of course, we have used radio using these electromagnetic waves. People like Marconi had patents for radio. In Spanish, a person named Cervera, also in Spain, a person named Cervera, etc. did this. So this is an illustration. This slide here is an illustration that you could take phenomena like electrical phenomena or magnetic phenomena described mathematically, and then you could, if you are Maxwell, you are able to actually come up with a set of equations that actually unify this, and that set of equations has predictions. Can you do something with these properties for life? And the answer is yes. So these are the main questions we will be attacking in the remaining half hour or so. And there will be, the questions are, can a generic mathematical physics and chemistry, high level, little detail, description of the properties of life be given? And the answer is going to be yes. And this is, of course, whatever, if you give the description, a description of life using some mathematical theory, then what you are, or some mathematical description, you need to solve that description, solve the equations. And if you solve the equations, you should see that the equations in their solutions display all the properties of living system. And there is such a system, and it's mathematics. It's powered by mathematics. It's not chemistry powering it. And then the next question is, in the sense of what you do in engineering, for example, in chemical engineering, could one use the above system as a guide in the laboratory synthesis of mimics of simple life? Far simpler than biochemistry, not using biochemistry at all. Or maybe even as the originator, you may even be able to get a chemistry that actually allows you to originate life in a very simple way, which is not, does not have all the complexity of biochemistry, but still, you would, if you saw a microscopic picture or a picture of these systems, you would deduce, or you would conclude that they were living. This is an implementation, as I mentioned earlier, of Bernal's notion of generalized or cosmic biology. That is to say, a biology which does not require biochemistry, but which has never been shown to exist until the experiments we're doing now are showing it. And we are really amazed that this can be done. So you could start representing living systems by thinking of the minimal physical-chemical properties that you need to include, and you need to make sure that you have chemistry. And in chemistry, you need a mechanism to transport the molecules so that reactions take place. And they react when they meet each other. So the mechanism that we will think of is diffusion and then reaction of the molecules. We will think you will need a minimal amount of 3 different substances, 2, one of which is the cell itself, the inside of the cell itself, the cytoplasm, as I would call it, and 1 for food, what the system eats. The system is an out of equilibrium. Living systems are out of equilibrium systems that that consume chemicals to operate. And then, of course, living systems produce waste. So you would need a minimum of these three objects, these three functions. And then you also would need an environment, which you will represent with an intrinsic component, which is a component which is intrinsically random. And randomness will operate in the feed. For example, today we have food, tomorrow we don't have food. So, and we don't know when it's going to come, and so on and so forth. So there is some randomness in the system, which needs to be incorporated. You can actually represent all of that mathematically, and then use the minimum mass action kinetics to incorporate waste and unify the four properties of life. And you come up with this set of equations. This set of equations are a diffusion equation, the last term and the left-hand side constitute the diffusion. There is a reaction in the first term in the left-hand side. There is autocatalysis, there is the feed, and so on and so forth. And the system needs some initial conditions or boundary conditions to actually start. You will see what happens when you supply the right initial conditions. If you're interested in this, you could look for my name in Physics of Life Reviews, and or the name of my collaborator in this paper, Alberto Pérez-Mercader, which came out in Physics of Life Reviews in 2022. And it's also an outgrowth of work that I did together with Lesmes, Felipe Lesmes, David Hochberg, who unfortunately is no longer with us, Federico Moran, and myself, which we published in Physical Regulators in 2003 and Physical Review E 2004. When you put these equations in the computer with the three components, with the three chemical species, and you give the appropriate boundary conditions, the system does this. This looks like a crazy dance, but what you can actually follow this in the computer, and let's say you follow one of those domains. You need to start a system. You do need to have a boundary condition, which is the presence of a gradient of free energy. That's very important. But if you do that and then you use those equations, you will see here a reproduction of the a presentation of what these systems do in terms of the cell division cycle. You can see how the mass and size grow. Okay. And you can see how the information in the system grows. And actually, you can do that and generate many generations. And you can see, for example, this is for 4 or 5 generations. We can do that up to 28 generations. You can see that the size represented by the green trace and the mass represented by the red trace, the red trace do follow something like this. This is extremely idealized, this presentation of the cell division cycle. And you can see that the information in arbitrary units, the information is characterized, minus H is characterized by the Gibbs free energy, the negative of the Gibbs, not free energy, the negative of the Gibbs entropy. And it's a form of the Shannon entropy. So with that, you then say, so I can imagine that I can see these equations, if I give it the right boundary conditions, generate systems like this, and then those systems actually execute the cell division cycle. So as it is well known for many of us from Spain, we would like to quote what Picasso said. Picasso said, everything you can imagine is real. Well, it depends on how much marijuana you have smoked, how much wine, good wine or bad wine you have drunk, and so on and so forth. That's, of course, it's a joke. But if you do it with the help of the scientific method, you can make it into a reality. And that is what we will do. We will take guidance from the solutions of these equations, just like Hertz took guidance from the solutions of Maxwell's equations, and we will take that guidance from solving these equations, and it will give us hints as to what to do. And the hint, it will provide us with a translation of the equations into a chemistry. Can we translate these ideas, these equations, into some chemical network that can boot up, just like a computer that starts by itself? It can boot up. and sustain an artificial, chemistry-operated living system in water, it has to do it autonomously, and it has to come from a completely disorganized state, a homogeneous state, which is a state of the highest entropy. For that, you need to solve two problems. And one of the problems is the concentration problem, otherwise known as the arithmetic demon. I'm not going to be having a lot of time to talk about this now, but if you are interested, there was somebody in Spain, in CSIC, his name was Ceratosa, who actually generalized some of the ideas put forward by Ireland and others in the 1990s, and he described the arithmetic demon, which is that when you do complicated chemistry, you need to do many reaction purifications and so on. Each of those has an efficiency of less than 1. So when you multiply many steps, which are less than 1, you multiply them through, so because they're independent steps, then you get something which is very low efficiency. So with life has solved that. And I will say in a minute how it has solved that. The same thing happens with the Maxwell demon, which is that you need to have some information somewhere, and you need to use that information to actually go from disorder to order. That is called the Maxwell demon, and it's a problem in information theory. How can we overcome these two major difficulties? Well, our computer simulations have shown, not simulation, solution to our equations has shown that you can overcome those difficulties by introducing a free energy gradient. And well, what are the boundary conditions? The boundary conditions are open permeable systems. So energy and information-driven self-assembly and encapsulation is something that in principle should be possible. So the first thing we need to do is to generate a localized gradient free energy and generate something like a vesicle. And what is the membrane material? Well, I thought a lot about that and for a long time, and I was not coming with too many interesting things until I learned that there is a method of polymerization, which is RAFT, reversible addition, fragmentation, chain transfer, polymerization, which is a form of controlled radical polymerization, living controlled radical polymerization, which allows you to generate chains which are to polymerize a material and generate chains which are of a homogeneous length, as opposed to traditional radical polymerization, which gives you chain lengths which are in the polymerization, chain lengths which are very different, and therefore you do have very broad distribution of the molecular weights, and that wouldn't help very much. Unbeknownst to us, in 2009, and probably a little earlier than that also, there was work by Pan and Juan and Sun and others, which appeared in macromolecular rapid communications, which showed that you could use RAFT polymerization by choosing where you have for example, a hydrophilic block, that is to say, a polymer that likes water, at which end you attach what is called a chain transfer agent, put it in a solution. All this previous work was in alcohols and so on, and then have a monomer which is dissolved in the solution, and then the hydrophilic tail will do this, will be elongated like that, and that hydrophilic tail that is elongated has here the chain transfer agent, which contains sulfur, which helps in the polymerization of these guys. And the following happens. I hope I don't. Yes, it's working. So you start the chemistry, the polymerization chemistry, by shining, for example, light. You could do use different initiators, and today even you don't even need to use PISA with RAFT. You could use other forms. And what happens is that the monomers, which are these red monomers which are around, will be polymerized, displace this chain transfer agent, and then these monomers can be chosen to generate, when they polymerize, a hydrophobic tail. So you have then an amphiphile, a molecule, a polymer, which contains, just like some lipids, contains a hydrophilic part and a hydrophobic part. These, if you increase, if this is in water and you increase the concentration then they are unstable and they will self-organize into micelles and so on that you will see. So in a little bit more detail, this is what happens. You start with some hydrophilic block, and then by raft you add monomers, and the monomers polymerize into this type of structure. Then they generate a diblock copolymer, 2 blocks, hydrophilic and hydrophobic. And then depending on the length of this hydrophobic block, the system will first make micelles or spheres, then the system may make elongated micelles, and it may make vesicles. And you can ask, but isn't that what could generate something that is similar to what the equations told us would do and would end up happening? And the answer is yes. So my collaborator and postdoc and friend, Jan Szymanski, and myself, we published a number of papers there. They're written around that time, and where we generalized this system from alcohol to water, and then we generated systems which were actually visible in the microscope because we wanted to see their evolution. So we polymerized, we ended up generating vesicles like this that we could see growing in the microscope, and we were super excited. And then part of this work appeared as the cover of Polymer Chemistry. And then we came up with a different system that we were able to tune much better, where we mixed in a small cuvette, 10 cubic centimeters, we mix water, in this case, hydroxypropyl methylalkylate monomer. We could also use acrylonitrile. We could use the PEG with the raft agent attached to it, containing sulfur. Ruthenium as a generator of radicals, helping in the generation of radicals, in the ruthenium in the form of tris-butyryl chloride. And then we would put it here in this reactor, just supply water to, this is a batch reactor. This is just to keep the water temperature constant. And we would withdraw a sample from time to time and follow the reaction, the details of the reaction. And here is what you get. You follow, you start with something which is transparent, such as this in this vial here. The reddish color has to do with the presence of ruthenium. And then as the light initiates the photochemistry, there is the beginning of the reactions to take place. And as the beginning of the reaction starts taking place, eventually the system accelerates, it becomes turbid, which means that there are supramolecular structures which are formed in the system, and then we study them. And what we found out is that you could see how the systems formed, and here is a video of what we discovered. This is a microscopic video, a video taken with a microscope. It's not a video that is the results of a computer simulation or anything like that. And then you can see there is light shining here at a stronger intensity. So you will see that these guys, which were, I'll show you a computer movie of this in a minute, but this is real. This is from the laboratory. And then you will see that these guys begin to grow. And as they grow, they move and they tumble to where there is more light. It is because the photochemistry is more effective here. And then you will see that as they tumble, they do something really interesting. Let's look at this guy. So he is growing. And as he grows, you can see that it's a structure which is about 10 microns, 10 to 15 microns. And then he collapsed. And when he collapsed, more of them began to appear next to him. So he's collapsing, squeezing out things from the interior, and then those things, spores, we call them, actually serve to generate more, because in this reaction medium, you have all the ingredients to actually be able to start the booting from scratch, from zero. And this is totally autopoietic. and from a homogeneous out of equilibrium mixture of a few non-biochemical molecules, including the fuels, for example, acrylonitrile or HPMA, we go to a phase-separated autobiotic ecosystem. If you look at the cycles of these guys, you see that the radius grows and then collapses, grows, collapses, grows, collapses. This is some of the first reports. In our laboratory, just in order to make sure that we're doing the things correctly, we have this sign at the door. Non-biological materials allowed, except for researchers. And this is the sign on our door. And it's important because we don't want to have any false results. I mean, false positives or anything like that. So it's totally abiotic, meaning a chemistry which is not biochemistry. It's organic chemistry, it's carbon chemistry, but it's not any biochemistry. And you can see, you can count the number of vesicles, as we, for example, have done in this communications chemistry paper with Cheyu, Lin, and Saikatla, and you count as time goes by, and you can see that there is an increase, which is a logistic increase, and of course this is in a batch, so it will start consuming the materials, and it will eventually entropy will eventually kill the system or reduce the possibilities of the system. This is we understand a lot of what goes on here, and we understand the details of what goes on here, how there is a transition from micelle to vesicle, and that transition from micelle to vesicle, we understand what happens in the interior, how the micelle begins to drink water, and how the drinking of the water leads to inverse micelles forming inside, and the inverse micelles then eventually couple and make a bilayer vesicle, and then the bilayer vesicle begins to grow, and when it begins to grow, if it grows at too fast a rate, then it generates pores, and there is cavitation, a solution of the Navier-Stokes equations, which are the ones that describe the evolution of the membrane, and then it contracts because of the elastic forces in the membrane, and then again does it over and over again. And this is some work that right now we are writing up with Cheyu, Sai, and myself. And let me show you in a little bit more detail. I have like 4 more minutes till I am able to finish. Let me show you this a movie by Janet Iwasa, which is work in progress, where you can see the molecules. You can see this is the hydrophilic tail. This is the ruthenium molecules. This is the chain transfer agent with 2 sulfurs there. And let's see what it does. So when there is no light, dark, nothing happens. The red are the monomers which are going to polymerize, and the light begins, the monomers begin to polymerize, and then you begin to see the growth. And then in order to make things easier, we have gone from this chemistry detailed view, to this stick and blob. So the blue still is the PG, polyethylene glycol, how they rearrange themselves and how they form structures which actually begin to develop defects. The defects make it break and collapse, and then they squirt out seeds, and those seeds become new vesicles. This is work in progress for this movie. And this is, we have shown in several places how this takes place, and we have also studied if these systems can be made to compete. So we have, because that's very fundamental in evolution, is the competitive exclusion principle. And we have put together systems. If you're interested, you can look at Cell Reports Physical Science in 2023, Katla and Lin and myself, and you can see that you can take 2 types of vesicles which contain for example, a catalyst, which likes the type of light that you have, and vesicles which don't have that catalyst, and the ones which have the catalyst, if there is food, or if there is less abundant food, they will, we should say here, less abundant food in this one. So they will grow and so on, but then they will start competing for resources. They will start competing for resources, and you can see that in the pictures. You can look at transmittance images telling us which ones contain zinc. Those are the ones that you see here in red, and the ones which don't contain. And eventually they overcome. You can actually track their growth and how they compete, how they grow when they are grown separately. The ones with the zinc tetraphenyl porphyrin, which is the catalyst, and without the tetraphenyl porphyrin, which is the other variety. And when they grow separately, they follow these paths. This is without the green, and with zinc tetraporphyrin is the ones in red. And when they grow together, they compete, and they compete and this guy cannot grow. The one without zinc TPP cannot grow the same way as the other one, and therefore eventually they actually decay. This is completely analogous to paramecium in a competition in paramecium species. This is a classical set of experiments to actually demonstrate the struggle for existence in the laboratory, and our bichos, we call them, biologically inspired, chemically operated systems, just do the same thing. And they compete. Here you have an image of how they grow together, and the ones which have the advantage eventually compete with the ones which don't have the advantage, and eventually displace the population, as shown in this objects. We can do much more than this. We can also, I don't have time to discuss all of that, of course. We can do much more than that. We can make systems. We can make systems that adapt. And this, you can look at the publication is in ACS, American Chemical Society Central Science, in 2021 with Sam Pierce and myself. And then in Nano Letters in this year, Sam Pierce, Chen Yu, and myself. And we can make actually systems that adapt, and in addition, systems which do not need to start with a macro raft agent. We can actually synthesize the macro raft agent from much simpler parts by combining click chemistry with PISA. And what we do see is that, like Stephen Jay Gould said in an editorial to the New York Times in 1996, when it was made public that life in quotes was in the Martian meteorite, and he didn't question the details of that, but what he said is the origin of life may be a virtually automatic consequence of carbon chemistry and the physics of self-organizing systems, given favorable environments and requisite inorganic constituents, which is precisely what we have done. And let me end by offering you a set of conclusions that are here. Natural systems share a set of 4 properties, and these properties are integrated in the universal cell cycle, and they are expressed today on Earth using biochemistry. In its early stages, life on Earth probably was far simpler. Biochemistry is very complex. There is a huge jump in complexity from regular chemistry to biochemistry and to life. So we asked, what does it take to make a self-footed, fully chemically operated, completely synthetic living system, something that behaves like life, but doesn't contain any biochemistry? And if we can do that, then biochemistry is sufficient, not necessary. So life properties can be unified and represented by a simple set of stochastic reaction diffusion equations. Then they are biochemistry independent. And when you solve those equations, you discover a set of boundary conditions that require a free energy gradient that lead to systems that actually behave like living systems. These systems with a free energy gradient would be vesicles or quasagrates. So the equations, we have found out a way in which the equations can be translated into photo PISA, occurring in a one-part chemical implementation, like a warm little pond, involving only a few simple compounds in water and light. They autonomously boot up. These simple molecules, 5 or 6 molecules, different classes of molecules. They autonomously boot up, that is start, as self-organized mice cells, and then the mice cells evolve, not in the Darwinian sense, but in the sense of changing with time, into a population, very important for biology, of biochemistry-free vesicles that they handle information, is conformational information that they have there. It's not polymeric information in the sense of DNA, but it's information, chemical information. They metabolize, they self-reproduce, they do the struggle for existence, they compete, and they also have inheritance, because the reproduction by spores contains heritable variation, which is in the spores that are put out. The polydispersity index is not 1. So there is a variation from small variations between different spores, but that enables heritable variation, which is key for the Darwinian evolution. We can also have adaptivity by enlarging complete PISA systems. And we conclude that life is a consequence of carbon chemistry and the physics of self-assembly or self-organization, whatever, either one will work. And it is what we have done is a form of Bernal's postulated generalized life or life beyond biochemistry. This has a huge number of implications for the construction of really synthetic living systems, connecting life to the interstellar medium chemistry. There are many possibilities for experimenting with these systems. Life in exoplanets, we predict how life in exoplanets could be in the universe, to better study in the laboratory the origin of life and the protocell era on the Earth, that is to say, the era before Luca, and to actually try and understand better the presence or not presence of life in the universe. These are my sponsors: Repsol, a Spanish company, our university, the Origins of Life Initiative, and the Center for Nanoscale Systems in Harvard, the Fundación Everest, and the National Science Foundation, which provides this wonderful facility for us. And with that, and no further ado, and apologizing for taking a few more minutes than you probably wanted to hear, here it is, my thanks. So I pass the floor to Maripaz and Francisla, and I am open to any questions that you may have. Thank you very much for your attention.